Assalamu alaikum. Now that the cell has produced messenger RNA, it's time to translate it into proteins. So in this lecture, we'll be talking about the process of translation and how it's regulated. So as always, main resource is this lecture, and you can refer to Cooper's chapter 9 as well. Now, the process of translation involves interaction of three different RNA molecules. And these are the transfer RNA, ribosomal RNA, and messenger RNA template. So this is the main molecule. The messenger RNA is the main molecule that allows us, or that is read and translated into a polypeptide. Now, ribosomal RNA molecules are important structural molecules, and they have functional role as well. They catalyze the reaction of connecting two amino acids to each other. That is formation of a peptide bond. And then we have the transfer RNA molecules. They are small RNA molecules that transfer amino acids to a growing polypeptide. Now these RNA molecules assemble in ribosomes or with the help of ribosomes, and these ribosomes are large structural objects, bodies that exist in cells, and they are the real factory of protein synthesis. So right within ribosomes, you have these ribosomal RNA molecules, and then you have the integration of messenger RNA template as well as the tRNA molecules um, uh, synthesizing polypeptides. Now, a very important point is that about decades ago, when the scientists started studying DNA and they realized that it is the functional hereditary molecule, they noticed that there is some sort of like collinearity between genes, messenger RNA molecules, and proteins. And the idea is that if you have a DNA, you would have exactly sort of like a replica of this DNA um, transcribed into messenger RNA. And the order of nucleotides in DNA resembles the order of nucleotides in the messenger RNA. And then you have this order right here that that is translated into proteins, although they're different. And here we're talking about nucleotides versus amino acids. But scientists have noticed that if there is a change at the beginning of the messenger RNA, or a mutation that is, there would be a change as well in the beginning of a polypeptide. If there is a mutation, a deletion, any type of change that occurs at the end of the messenger RNA, this would also be translated into a change at the end of the polypeptide. So there is this collinearity of genes, messenger RNAs, and proteins, and that clarifies the idea that you have genes basically that are read, transcribed into messenger RNA, and this messenger RNA is read and translated into amino acids in the same order. So that was like an important point at that time. Now, the messenger RNA, when it is translated into proteins, it is not read nucleotide by nucleotide. Rather, it is read in a group of three nucleotides. And these three nucleotides are known as codons. So we say that mRNA is read by tRNA molecules in triplets. So it is really the tRNA molecule that forms a complex with the ribosome that is responsible for reading the codons in the messenger RNA. So scientists tried to translate these codons, these triplets, into amino acids. They did a lot of experiments, and they found that each codon, each triplet, is specific for a certain amino acid. Now this table is not for you to memorize, okay? But just to show you what each codon represents. So right here, for example, let's say that you have a codon that is CAU. Okay, so the first letter is C. That we're talking here about the codon in the messenger RNA itself. Okay, so CAU. So the first letter is C. Okay, now the second letter you go up here and you say, aha, it's A, and then you have uh, to stick to this box right here. So they intersect right here. Okay, so CAU, and you look at the third letter, so it is U right here. So that's CAU, and CAU is translated into an amino acid known as histidine. Okay, and it is abbreviated as HIS. Let's look at another example right here. So let's say we are looking at GCA. Okay, so the first letter is G. Okay, so you have to to restrict yourself to these four boxes right here. So we said GC. What did I say? GCA. Let's see, GCA. Okay, so you have to go to GC. That's the second letter. So now you are restricted to this box right here, GCA, and notice that GCA is translated into another amino acid known as alanine, and it is abbreviated as ALA. Okay, so this is how you know what each codon is translated into. Now, there are four codons that I encircled, sort of like inboxed. And these are AUG, then you have UAA, UAG, and UGA. Now these four codons, you have to memorize them. Now you don't have to memorize any of the other codons, and there are 64 of them, by the way, but you have to memorize these four. Now the reason is that they have functional importance. So for example, AUG is the codon for methionine, another amino acid, methionine, and methionine is encoded by only one codon, and that is AUG. That's it. Now the reason why this codon is important, because this is the first codon that is translated. Okay, and we will get to that. Now notice that all of the other amino acids can be encoded by two or more codons. Like for example, you have this amino acid right here, proline, or pro, abbreviated as pro. It can be encoded by any of these four codons: CCU, CCC, CCA, CCG, and so on. But methionine is the only amino acid that is encoded by a single codon, which is AUG. Then we have these three codons, UAA, UAG, and UGA. Now these three codons are not represented uh, by any amino acid, and they encode a stop codon. That is, this is where translation terminates. That's where it ends. It ends whenever you get to this codon or this codon or that codon. Now, there are certain features and characteristics of this table, the generic codon as we call it. And some of these features is that there are, as I said, 64 possible codons, different combinations of these three letters, because you have four. There are four possibilities for the first letter. 4 letters for the second position of the codon, and 4, of course, for the third. So it's 4 times 4 times 4, and that is 64. Now we said that 61 of them encode amino acids, but 3 of them are known as stop codons, UAA, UAG, UGA, and these are the codons that terminate translation. Now, the messenger RNA is read from the 5' end to the 3' end, and proteins are synthesized from the amino terminus to the carboxy terminus. Okay, so we'll get to that as well. Now, this genetic codon table is really a universal table. Now, there are, of course, exceptions. As always, they exist in bacteria as well as mitochondria, since they have their own DNA and they have their own transfer RNAs and genes and whatever. Okay, so there are some exceptions, but overall, it is universal. Now, let's look at the transfer RNA first. Now, transfer RNA molecules are short, single-stranded RNA molecules, and they are about 80 bases long. So they have their own 5' starting point, and they have a 3' end point as well. Now, there is a consensus sequence at the end of the tRNA molecule, at the 3' end of the tRNA molecule, and it is CCA. Now, to the A, you have tRNA molecules attached to amino acids, or you have amino acids attached to the A of tRNA molecules. Now, this tRNA molecule that has an amino acid attached to it is known as a charged tRNA. Otherwise, it's called uncharged. Now, attachment of amino acids to tRNA molecules is catalyzed by enzymes known as amino acid tRNA synthetases. Now, what these enzymes do is that they recognize the tRNA, and there are multiple synthetases, by the way. Now, what these enzymes do is that they look at the tRNA molecule, they look at certain sequences within the tRNA itself, particularly a 3 nucleotide sequence that exists like at the bottom of the tRNA molecule, and it is known as the anticodon. Okay? And according to the anticodon, as well as other sequences within the tRNA molecules, but mainly the anticodon, you have a specific amino acid that is attached to that A at the 3' end. Now, the structure of the tRNA molecule is quite unique. It is exemplified according to this structure right here. So it looks like what we say a clover leaf. So this is a clover leaf right here. And the reason why it has this structure is because there are internal hydrogen bonds that are formed between complementary bases within the tRNA molecule. Something interesting about tRNA molecules as well is that they may contain inosine instead of adenine. So you have modification of the adenine within the tRNA molecule, and it gets converted into an inosine. Now, so what's important about anticodon? It is the part of the tRNA molecule that recognizes the codon, the genetic codon, the triplet that I just told you about. Okay, so in the messenger RNA, for example, let's say that you have this sequence AUA. Now you have this complementary base pairing between the tRNA and the messenger RNA molecule. Okay, now so once you have this base pairing, this means that you have the right tRNA that is bound to the codon, and then you have transfer of this amino acid into the growing polypeptide chain. Now notice something, that this base pairing between the tRNA and the messenger RNA follows the anti-parallel rule that we just talked about. So messenger RNA is 5' to 3'. Notice that the tRNA is anti-parallel. It's 5' to 3'. So pay attention to that. Now, translation is really highly accurate. And there are two reasons for why translation is accurate. The first reason is that you have this accurate association of amino acids to tRNA molecules by these enzymes, by the amino acid tRNA synthetases. Okay, so these enzymes are really accurate, so they do not mess up. They do not read an anticodon and insert a different amino acid. No, they are accurate. Now, the other thing is that you have to have this correct base pairing between the tRNA anticodon and the messenger RNA codon. Okay, so that's the second reason. However, there is something known as wobble base pairing. What wobble base pairing is that there is some sort of flexibility in base pairing, particularly when it comes to the third nucleotide of the codon or the first nucleotide of the tRNA molecule, of the anticodon of the tRNA molecule. So there's flexibility. So the first nucleotide within the codon has to pair correctly to the anticodon. Same thing with this second nucleotide. But there is some sort of flexibility when it comes to interaction between or base pairing between the third nucleotide of a codon with the anticodon. And this flexibility is important because what it does is that it acts as a buffer, meaning that what it does is that it protects it protects cells and proteins from mutations. Okay, and I'll show you an example right now. So basically, the base pairing between the codon and the anticodon is really not let's say very specific when it comes to the third nucleotide of a codon, and it is related to the degeneracy of the codon of the genetic codon. Degeneracy means that you have some sort of like level of dirty business between codons and anticodons. Now this degeneracy of the genetic codon, or filthiness of the genetic codon, is very specific to the GU base pairing. So instead of having a GC base pairing, you have a GU base pairing. Okay, so right here you have normally you should have GC base pairing, but you can have this GU base pairing. Now, what do I mean by saying that it acts as a buffer against mutations? Well, the thing is, if you have a change in the third nucleotide of a codon, for many of these codons, nothing would happen, right? So basically, if you change a U to a C in a codon in the messenger RNA, you still code for the same amino acid, which is serine. Okay, so if you change U to an A, well, same thing. Okay, now notice right here, if you change U to C, nothing happens as well. So you still have this pairing between the tRNA and the messenger RNA. So nothing happens. You still have the same amino acid. Now, what's really interesting as well is that if you change a U to an A, you do change the amino acid. okay, from one to the other. But what's interesting is that both of these amino acids, as we will learn later on in biochemistry, both of them are hydrophobic. So basically the structure of the protein may not change that much. So we're not changing an amino acid from a hydrophobic amino acid into a positively charged hydrophilic amino acid. Totally different. No, no, no. So that would affect protein structure. But by changing an amino acid into another one, having the same overall chemical characteristics, the structure and function of proteins may be maintained. So this is sort of like, so by having this wobble based pairing, there is this protection level of protection against mutations. So let's talk about the second component in the process of protein synthesis, the second player, and that is ribosomes. Now, cells need a lot of ribosomes because they are the site of protein synthesis. They are the factory for protein synthesis. And there's a lot of protein synthesis going on in cells, especially healthy cells. So for example, E. coli contain about 20,000 ribosomes per one cell. And if you dry out cells and you measure the weight of the solid material, 25% of this solid material is basically ribosomes. Rapidly growing mammalian cells contain about 10 million ribosomes, so that's a lot. Okay, so there's a lot of production of ribosomal RNA inside these cells, and that's why, you know, if you remember when I talked about northern blotting, and I said that you really see a smear. Okay, you don't see distinct bands except for ribosomal RNA bands. There are two bands. Actually, there are three bands in bacterial cells. So anyhow, and these ribosomes, they are composed of a large ribosomal subunit and a small ribosomal subunit as well. Now, the large ribosomal subunit is the place where you have the formation of the peptide bond, that is the bond between two amino acids. So it is responsible for the synthesis of the polypeptide. Another point that is important is that the messenger RNA is read from the 5' end to the 3' end as well. Okay, so we'll talk about that as well in a few seconds. So whether we're talking about prokaryotic cells or eukaryotic cells, they have the sort of like quote unquote identical ribosomal subunits and identical ribosomal RNA molecules. They even have the same structure, and that tells you something about their significance. That is, they haven't changed much throughout millions of years of our again, quote unquote, evolution, quote unquote, creation. So if you look at prokaryotic cells, if you look at the ribosomes of prokaryotic cells, they are composed again of two subunits, a large ribosomal subunit that has a weight of 50 S. S is the unit of measurement of the weight of proteins or objects inside cells, and it stands for Svedric. Okay, it's like meter or something, or kilograms or grams or whatever. Okay, Svedric. And a small ribosomal subunit that has a weight of 30 S. Now, the large ribosomal subunit inside bacteria are composed of two types of ribosomal RNA molecules, and these are the 23S and the 5S. And there are many proteins. Now, if you look at the small ribosomal subunit, it is composed of 16S ribosomal RNA as well as 21 proteins. I don't care about these numbers, 34 and 21, by the way. I care about
the type of the ribosomal RNA molecules. Now, if you look at eukaryotic cells, again, you have large ribosomal subunit and small ribosomal subunit, and the large ribosomal subunit has a weight of 60s, and the small ribosomal subunit has a weight of 40s. The large ribosomal subunit is composed of 3 ribosomal RNA molecules, 28s, 5.8s, and 5s ribosomal RNAs. The small ribosomal subunit is composed of plus 45 proteins, by the way. The small ribosomal subunit is composed of the 18s ribosomal RNA as well as 30 proteins. Again, I don't care about 45 and 30. I only care about the type of ribosomal RNA molecules. Now, someone would say, well, wait a second. Look here. The size or the weight of the ribosome in eukaryotic cells is 80s, whereas for prokaryotic cells it is 70s. So why is it that it is not 100s? Because that's right, the sum of 60 and 40. Well, the reason is that the unit of Svedrick does not depend on really actual weight. It also depends on the shape of the molecule. So if you have only the large ribosome subunit by itself, it has this weight because you have to consider the shape. Now, once it forms a complex with the 40As, it will have a different shape, okay, and as a result, a different Svedrick unit. Same thing with the prokaryotic ribosome, by the way. Okay, so it is 70 instead of 80. Now, let's talk about the ribosomal RNA molecules. Now, these ribosomal RNA molecules are synthesized by RNA polymerase 1. Okay, we're talking here about eukaryotic cells, by the way, not prokaryotic cells. Okay, so remember we said that there are 3 RNA polymerases in human cells. We have RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. RNA polymerase 2 is responsible for synthesizing the messenger RNA, whereas RNA polymerase 1 is responsible for synthesizing ribosomal RNA molecules, specifically the 18S, the 5.8S, and the 28S. Whereas the 5S ribosomal RNA molecule is synthesized by RNA polymerase 3. Okay, so RNA polymerase 1 is responsible for synthesizing 18S, 5.8S, and 28S. Okay, the thing about human ribosomal RNA molecules is that there are multiple genes and they are tandemly ordered. So it's one after the other in a certain location in our genome. So you have synthesis of identical ribosomal RNA molecules from multiple genes. And the reason is that to produce as much ribosomal RNA molecules as possible in a very short time. Same thing for histones, by the way. There are multiple histone genes. So you have multiple histone 1 genes and 2A, 2B, and so on, because cells need a lot of it, so they duplicated these genes. So these 3 ribosomal RNA molecules are produced from these genes, and they are produced as one primary transcript, or one primary RNA molecule. And then it gets processed. So this RNA molecule is cleaved specifically into the 3 RNA molecules, ribosomal RNA molecules, the 18S, the 5.8S, and the 28S. Okay. So this is known as post-transcriptional processing. Okay, so there we have transcriptional control, just like the lac operon, for example, synthesis or transcription of certain genes in human cells being controlled by enhancers and promoter proximate elements, and so on. And then we have post-transcriptional regulation. So this is considered post-transcriptional regulation processing or cleavage. Now the 18S is associated with the small ribosomal subunit. Now the 5.8 and the 28S rRNA molecules associate with the large ribosomal subunit. On the other hand, now we have the 5S ribosomal RNA, which is produced by a different gene, by the RNA polymerase III, and it associates with the large ribosomal subunit as well. Now, as we will see in a few minutes, ribosomes associate with the messenger RNA template as well as the transfer RNA, and you have the initiation of protein synthesis. Now, the question is, what is the role of the ribosomal RNA molecules? And amazingly, what ribosomal RNA molecules do is that they appear to function as enzymes. They catalyze the reactions of associating, linking amino acids to each other, and synthesis of polypeptides. So, and this was done by nice experiments where proteins were removed from ribosomes, and even if 90% of proteins were removed, still ribosomes were able to synthesize polypeptides. Okay, so if you look at the structure of these ribosomal subunits, they, by the way, they are located in the sites where proteins are, or sorry, when peptides or polypeptides are synthesized. And this is another evidence for the role of ribosomal RNA in protein synthesis. Looking closely at ribosomes, there are three rooms, there are three chambers inside the large ribosomal subunit. That's why I put this in here, because I love Harry Potter's movies. Now, so there are three chambers. Okay, you have a place where you have that is a chamber or a room, a site where you have the synthesis of the polypeptide, the formation of the peptide bond. You have a place where you have the second tRNA molecule added, and you have an exit chamber as well, here where you have the empty tRNA placed, and then it gets removed from ribosomes, as we will see in a few seconds. Okay. So let's talk about the process of translation. Again, it seems that there are, well, we can divide the translation into three stages. We have initiation, elongation, and termination. Now, this is not only done for convenience. It's nice, of course, to simplify things, but because we have certain proteins that are responsible for initiation, others for elongation, and we have a third class of proteins that are responsible for terminating translation. The direction of synthesis, or the direction of reading messenger RNA, is 5' to 3', and proteins are synthesized from the amino terminus to the carboxy terminus. Okay, so the idea here is that if you look at an amino acid, it has an amino group and a carboxyl group, and the second amino acid, or the incoming amino acid, is added to the carboxyl group of the first amino acid. Okay, so and so on. So the just like having the 5' end in DNA or RNA untouched, same thing with polypeptides. The amino group of the very first amino acid is not touched, and any additional amino acid would be added to the carboxyl group, or to the carboxyl terminus of polypeptides. So translation starts at a specific site, which is AUG. So AUG, as I said before, it encodes methionine. Okay. Now, So translation does not start at the very codon of messenger RNA molecules. No, it starts at at an AUG that comes right after the 5' end. Now notice that for the messenger RNA it is AUG 5' to 3'. Notice that the complementary tRNA or the tRNA must have a anticodon of CAU starting at the 5' end. Okay, and this happens before the complete assembly of ribosomes. So what you have in here is the small ribosomal subunit binding or associating with the messenger RNA, and the tRNA, the first tRNA that has a methionine associated with it, and it has the anticodon of CAU. It binds or it base pairs with the AUG. This is followed by hydrolysis of GTP to GDP, allowing for the binding or association of the large ribosomal subunit to the whole complex. Now, since we're not really starting at the very codon of the messenger RNA and we're looking at the AUG that comes right after, the 5' end, it means that there is a region that is not translated into a peptide, and this region is known as the untranslated region. And you have and it's known specifically the 5' untranslated region. Okay, so right here you have the cap, okay, and that's in eukaryotic cells, human cells of course, and you have a region that is not translated. Now there's also another untranslated region at the 3' end. Here you have the poly A tail. So you have another region that is known as the 3' untranslated region as well. Now, remember that some bacterial genes are polycistronic, meaning again that you can produce different polypeptides from different regions of the same messenger RNA. So how do ribosomes know that there is a region right here that needs to be translated, another that needs to be translated as well, and a third one? There is an AUG for the first one, by the way, and there's another AUG in the second one, and there's a third AUG for the third region, for the third cistron. So does that mean that there is no AUG in the middle of proteins? No, there is. So how do bacterial cells know that they should start from this AUG, and this AUG? Now, remember as well that for eukaryotic messenger RNA, genes or messenger RNA molecules are monocistronic. Again, how do cells know that they should start at this AUG, not one that exists right here? Or maybe there's an AUG right here as well. How do cells know that they should start at this point, at this AUG? So for prokaryotic cells, there is a region whereby cells know that they should start at the AUG that comes right after it. And this region is known as the Shine-Dalgarno sequence. Shine-Dalgarno, not Delgarno. Okay. Now this sequence it's a consensus sequence, by the way, and I don't want you to memorize it. Okay. And it's complementary to part of the 16S ribosomal RNA molecule. Now this ribosomal RNA is part of the small ribosomal subunit. Now, once you have association of the 16S ribosomal RNA, which is part of the small ribosomal subunit, then translation starts at the AUG that comes right after it. So if there is an AUG right here, it's not considered as the starting point for translation. Rather, it's the AUG that comes right after the Shine-Dalgarno sequence. So there is actually a Shine-Dalgarno sequence right here there is another one right there, and there is a third one right here. That's in case of the lac operon. Okay, and it's the AUG that comes after the Shine-Dalgarno sequence that is considered the starting point for translation. And there's another AUG right here and a third right there, and that's how cells know that they should start at these AUG codons, not any other AUG. But how about eukaryotic cells? Well, things again are different. Now what happens is that small ribosomal subunits look at the cap at the 5' end, and then they look at the AUG that comes right after it. Okay, so again, there is no specific Shine-Dalgarno sequence for many of the eukaryotic genes. So in eukaryotic cells, you have a group of initiation factors, and these are known as E for eukaryotic, initiation factor for proteins. So it's a set of proteins. And what these do is that they bind to the cap, okay, and they help associate the cap to the poly-egg tail. Remember how I said in the first lecture, or in the transcription lecture, that the poly-A tail is important for translation? And someone would say, well, wait a second, the poly-A tail is at the end of the messenger RNA, the part of the messenger RNA that is not translated. How is it important? Well, it's important because what happens is that you have these proteins, particularly you have the initiation factor for G, and you have another one, initiation factor for E. They form a complex with a poly-A binding protein, okay, and this helps forming the complex that is necessary for initiating translation. Okay, so it is sort of like a mechanism to ensure that the messenger RNA is fully processed, fully and accurately processed. Okay, of course you have other proteins, but I don't care about these. Just know that here we have a complex of initiation factor for proteins, including G and E. All right. So this is true for many genes, but there are some genes that do have something that look like a Shine-Dalgarno sequence. It's a different sequence. Okay, and it's known as the internal ribosome entry site. Now, so there is a site that comes right before the AUG, so you can consider it like upstream of the starting AUG codon, and it is recognized by initiation factor for G as well. Okay, and it can associate with other proteins as well. So it helps in the formation of the initiation complex, the small ribosomal subunit with all of the other proteins. Okay, and then you have translation after entry of the large ribosomal subunit. Then you can have translation going on at the AUG that comes right after the IRS sequence. Now, the first amino acid that is added is methionine, but there is a little difference between prokaryotic cells and eukaryotic cells, and that is in prokaryotic cells, in most bacterial cells, the first methionine is not just a methionine amino acid, it's rather a modified methionine known as formyl methionine. So here you have, this is methionine right there, and you have the methionine modified by the addition of a formyl group. Now, Then you have, after initiation, you have elongation and you have termination again. So these are the three stages. Now, as I said, as you noticed before, so initiation requires initiation factors, specific proteins. Same thing with elongation and termination as well. Okay, so what you have in here again, you have formation of this complex on the messenger RNA that I told you about. It involves, or it contains, initiation factor 4G as well as 4E. You also have a number of initiation factors that associate with the small ribosomal subunit and the tRNA, the initiating tRNA that carries the methionine and has the anticodon of CAU. So you have this association of this complex, which associates with the messenger RNA. And now you have all of these components: the messenger RNA, the small ribosomal subunit, the tRNA that binds to the AUG, and then the king comes in, the large ribosomal subunit. It associates with the tRNA and the small ribosomal subunit and the messenger RNA. Now note that all of these initiation factors are then removed. Okay. There's one initiation factor that I want to specify in addition to initiation factor 4G and 4E, and this initiation factor is number 2. Now this initiation factor is bound to GTP. Okay, so it's a source of energy, and it is responsible for bringing the first tRNA to the small ribosomal subunit. And once it does that, GTP is hydrolyzed, so a phosphate is released. Now this initiation factor is now inactive, and it has to be reactivated, and it gets reactivated by replacing GDP by another molecule of GTP. So what happens in here is not that you have addition of a phosphate to GDP, rather GDP is released and GTP comes in and it binds to initiation factor 2. And this requires another initiation factor, by the way, and it is known as initiation factor 2B. Okay, so this one here is different than this one right there. So what happens then is that this initiation factor, number 2, okay, is now active. Okay, and it can bind to another tRNA molecule, another initiating tRNA molecule that has a methionine. And you can have another messenger RNA primed for translation. So now we go into elongation, and elongation requires a number of proteins known as elongation factors, okay, EF, eukaryotic elongation factors. And what these do, particularly a elongation factor 1 alpha, is that it brings the second tRNA to the A chamber, to the A site. Now you have these two tRNA molecules next to each other. Then you have the peptide bond formation, and this peptide bond formation requires ribosomal RNA molecules. Okay, and what these do is that you have the formation of the peptide bond right here between these two amino acids, and the methionine, okay, it gets transferred to the amino acid that is on this tRNA molecule. Okay, so what happens then is that the ribosome, the whole ribosomal subunit, large and small ribosomal subunits, they get translocated. That is, they move one codon further. And this requires an elongation factor known as elongation factor 2. Okay, now you have this tRNA molecule placed in the E site, E chamber, which means exit, and it gets released. It gets kicked out of ribosome, and you have the second tRNA that, the second to this one right here, is now its place in the P chamber. And you have a third tRNA coming in, and it has an anticodon that is complementary to this codon in the messenger RNA. Okay, and again you have a transfer of two amino acids. You have transfer, yes, of the two amino acids to this one right here that is attached to the third tRNA molecule. Now you have three amino acids. Okay, ribosome attached to this tRNA. This one right here is empty. And you have translocation of ribosomes. Now you have a tRNA that has three amino acids, and this one is empty. It gets kicked out, and you have a fourth tRNA coming in. And then you have transfer, or you have peptide formation of the three amino acids to this to the tRNA that has a single amino acid attached to it in the A chamber. Now you have four. Okay, this one gets emptied, gets kicked out, and so on. So you have synthesis of polypeptide by adding one amino acid at a time from the carboxyl terminus of this peptide. Okay, so note that this right here is the very first amino acid. This one here is the methionine, and it gets untouched again. So anytime we want to add an amino acid, we add it to the carboxyl terminus, to the last amino acid that is added. Okay, so that's elongation. So that's how it goes. And so on. So you have this continuous movement, a continuous synthesis of the polypeptides. This is again the methionine, right? The second amino acid, the third amino acid. Here you have the amino terminus, and right here this is the carboxyl terminus of the polypeptide, and this whole thing is transferred, by the way, to this amino acid. You have the formation of the peptide bond between this amino acid and this one right here. Okay, and then you have translocation and so on, and the uncharged tRNA is kicked out. So let's look at this nice animation. Right here you have synthesis of the polypeptide. Right here, that's the elongating polypeptide. You have translocation of ribosomes. Now someone would say, well, what does move exactly? Is it the messenger RNA or the ribosome? Well, it is the ribosome. Okay, so but you can think of it in a different way. You can think of it that the ribosome is like sitting on the messenger RNA, and it keeps on pulling it underneath it. So it goes like this anyhow, and then you have termination. Now, this continues, by the way, showing synthesis of a membrane protein, so you can watch it at your own time. All right, so what happens again is that during elongation, the amino acid is added. All amino acids are added one by one to the carboxyl terminus of the polypeptide. So you have attachment, you have a peptide bond formation between the carboxyl terminus of the peptide and the incoming amino acid, right? So that's how it is. That's how it goes. We'll talk about this in the biochemistry course next semester, inshallah. And then we get to termination. Now, termination, remember I said that there are three codons that specify termination, and these are UAA, UAG, and UGA. These are the stop codons. Okay, now do not get them confused with the poly A signal, the termination signal for transcription. Here we are talking about translation, initiation of translation. It's not a start. Site, as in transcription. It's known as the start codon, AUG. Now for termination, we're not talking about the poly A signal. We're talking about a stop codon, and there are three of them: UAA, UAG, and UGA. There are no tRNA molecules that can recognize these three codons. Rather, you have proteins known as release factors. Okay, so there are a number of proteins. What they do is that they can recognize these stop codons. They bind to the A chamber and they release the whole complex. They dissociate the whole complex, and here you have termination of translation, and the polypeptide is now released. Right. So there's something important, an important difference between prokaryotic cells and eukaryotic cells, and that is in prokaryotic cells, translation and transcription are coupled in space and time. In other words, at the same time that you're having synthesis of messenger RNA from DNA, at the same time that you have transcription, you have ribosomes sitting on the messenger RNA and synthesizing proteins or polypeptides. So transcription and translation are coupled in space and time because they happen at the same place at the same time. On the other hand, in eukaryotic cells, they are not. You have transcription going on in one place and you have translation happening in a different place. Transcription takes place in the nucleus and translation takes place in the cytoplasm. Now the reason for that is that there is no nucleus in prokaryotic cells. There is no this nuclear membrane that separates the two from each other. That's why they do not happen at the same time at the same place. Now the other reason as well is that messenger RNA molecules in eukaryotic cells must get processed. They have to be capped. They have to be poly-A adenylated, and they have to be spliced. And then they get transported out of the nucleus once fully processed into the cytoplasm, and ribosomes cannot start synthesizing polypeptides until they recognize the cap. And they ensure that there is a poly-A tail as well. So in prokaryotic cells, you have the formation of polysomes, which I like to call polyribosomes, right? Remember, I showed you a picture of that, but it seems that the image that I showed you before is actually for only multiple synthesis reactions for messenger RNA molecules, not necessarily ribosomes synthesizing proteins. Okay, but in prokaryotic cells, what you have in here is you have a messenger RNA and multiple ribosomes doing protein synthesis or polypeptide synthesis. Okay, so here you have the DNA right here, this one here, and here you can look closely, you have the messenger RNA right there, and you have multiple ribosomes synthesizing polypeptides from this one single messenger RNA molecule. Now here you have another messenger RNA molecule and you have ribosomes doing polypeptide synthesis. Okay, so each one of these dots is basically a ribosome. We see the DNA, we see the messenger RNA, but by the way, we see the ribosomes, but we cannot see the polypeptide in here, in this image right here. Okay, so in eukaryotic cells, you can have one messenger RNA with multiple ribosomes doing synthesis. Okay, in prokaryotic cells, on the other hand, you can see DNA and you can see multiple RNA molecules synthesized from the same gene, from the same DNA, and you can see multiple ribosomes sitting on each mRNA molecule and doing protein synthesis or polypeptide synthesis. By the way, why do I keep saying polypeptide instead of protein and make this mistake? Because once you have a polypeptide, then you have the folding or the formation of a three-dimensional structure of a functional molecule known as a protein. So the difference between a polypeptide and a protein is that a polypeptide is basically just a stretch of amino acids, but for proteins, and it's not functional, whereas for proteins it's a three-dimensional molecule. It's a molecule with a specific three-dimensional structure, and it is functional as well. Now, so in eukaryotic cells, again, you have one single messenger RNA, and you have multiple ribosomes doing synthesis at the same time. So this is a polyribosomal structure. So everything has to be regulated, including translation. So in the next few slides, I'm going to talk about different modes or mechanisms of regulation of translation. So the first mechanism is basically regulating global translation. That is, in other words, all types of protein synthesis. So it's either that proteins are synthesized or not. Okay. So what happens in cells is that, remember I said that in order for translation to proceed, you have to have initiation factor to reactivate it, regenerate it. So whenever this initiation factor 2, whenever it binds to the initiating tRNA and it brings it to the ribosome, you have this hydrolysis of GTP to GDP. We already talked about that. Now, in order to allow for this initiation factor to restart, or to restart another round of translation, GDP must be replaced by GTP. Okay, so GTP comes in, it kicks out GDP, and now you have an active initiation factor 2. And it can again initiate another round of translation, like I said. Now, this also requires help from another initiation factor known as initiation factor 2b. So what happens is that if cells are under stress, or if there is no growth factor, that is, in other words, cells are starved, there are not enough nutrients for cells, cells are not in a position to start growing, and they have to preserve nutrients and energy, what they do is that they shut down translation altogether, simply by phosphorylating initiation factor 2. So whenever, by a kinase, okay? So whenever this protein is phosphorylated, you cannot regenerate active initiation factor 2. In other words, GDP is not released, and GTP does not bind. Now, same thing, by the way, for initiation factor 2b. It gets phosphorylated and it cannot function anymore. So translation altogether is blocked. Now, this global regulation of translation also works for reticulocytes. So reticulocytes are immature erythrocytes. So they are the precursors of erythrocytes. Erythrocytes are red blood cells. Now, these erythrocytes are full of hemoglobin molecules. Hemoglobin is the carrier of oxygen in red blood cells, and there are millions of hemoglobin molecules inside these cells. Now, so in reticulocytes, their only mission is to synthesize as much hemoglobin as possible. Of course, everything is under control. Now, hemoglobin is composed of two things. You have the protein itself, which is known as globin, and you have the heme molecule. Now, heme contains iron, and it is the oxygen carrier. And we'll talk about hemoglobin a lot in the biochemistry course. Well, sort of. So anyhow, so you have two things. You have the globin protein and you have heme. In case there is not enough heme inside cells, in reticulocytes that is, then there is no need to synthesize globin. Okay, it makes sense. So synthesis of globin is balanced or is controlled by how much heme there is inside these cells. So global translation is either initiated or activated, or it gets blocked. And this is done again by regulating the activity or the regeneration of initiation factor 2. So if there's adequate heme inside cells, you can have this exchange of GDP by GTP. Initiation factor 2 is activated. It can bring the initiating tRNA that has methionine attached to it. It can bring it to the small ribosomal subunit, as I described before. Now, in case there is no adequate heme, then in this case you have activation of the kinase, the enzyme that phosphorylates initiation factor 2, and in this case GDP cannot be released and it cannot be replaced by GTP. So translation as a whole is blocked. Also, another mechanism of regulation of translation, if you remember, we talked about ferritin before, right? And we talked about the stability of the transferrin receptor messenger RNA being controlled by how much iron there is inside cells. Well, the same is true for ferritin, except that for ferritin, it's translation that is controlled. So not stability of the messenger RNA. So what happens here is that if there's adequate iron, you have the iron response element or the iron regulatory protein. It binds to iron and gets released from the messenger RNA. So you have this formation of the whole initiating complex of translation, and it brings in the small ribosomal subunit, and ferritin messenger RNA can be translated. In case iron level inside cells is really scarce, there isn't enough iron inside cells, then the iron regulatory protein is not bound to iron because there isn't iron. Okay, and this protein binds to the element that I talked about before at the 5' end of the ferritin messenger RNA, and this prevents the formation of binding of the ribosome to the 5' end of this messenger RNA. It prevents the formation of the complex with the poly A tail, and there would be no translation of the ferritin messenger RNA. Now, another mechanism is by regulating initiation factor 4E. Now remember, this is the protein that links the poly A tail with the cap. Okay, so what happens here basically is that there is a protein known as that is known as initiation factor 4E binding protein. Okay, now this protein binds to the initiation factor 4E, and it prevents the formation of this whole complex. Okay, now what happens is that if there are growth factors surrounding cells, and that indicates this is a signal that the environment is nourished, there are enough nutrients, the situation is suitable for cells to grow and synthesize proteins, everything looks good. So if there are growth factors, what happens is that the binding protein, the 4E binding protein, gets phosphorylated, and once phosphorylated, it gets released, and you have formation of this whole complex. And now the small ribosomal subunit can bind to the 5' end of the messenger RNA, and translation can be initiated. And here is another mechanism as well. This is microRNA. Now, microRNA molecules are synthesized by RNA polymerase II, and they are synthesized into single-stranded primary miRNA transcripts. And what happens is that it gets processed by cleavage, forming a double-stranded microRNA. One of these strands, either one of them, can be degraded, and there is a protein complex known as RISC. It binds to the other strand, and this other strand can bind to the 3' end to the 3' untranslated region of messenger RNA molecules. So there has to be complementarity between the microRNA and the messenger RNA. Okay, particularly again at the 3' untranslated region of messenger RNA. Now, when you have this complex right here, when you have this hybridization taking place between the messenger RNA and microRNA, this prevents the formation of the connection, the link between the poly A tail and the proteins at the cap. Okay, and that either, well, there are two possibilities here. Either that translation is inhibited, or the messenger RNA itself, this whole thing, gets degraded. And this is again decided by cells, and there are other mechanisms, and we're not gonna go into detail. So this is what microRNA molecules do, and now we have discovered thousands of microRNA molecules in human cells, and they do a lot of work. They target a lot of messenger RNA molecules, and it's really a sort of like a well-coordinated mechanism of regulation of translation. All right, we're getting to the end of translation. So basically, you have synthesis of proteins. Now, proteins can be regulated as well, and we'll talk about different modes and mechanisms of regulation of proteins, and we're gonna keep that for the biochemistry course. But once you have proteins, they do not last forever. They can undergo turnover. That is, in other words, a cycle of degradation, synthesis, and so on. Okay, so when a protein is turned over, it means that it gets degraded. Now, so what happens here is that once cells have proteins and they do not need these proteins anymore, these proteins are labeled by small peptide molecules known as ubiquitins. This process is known as ubiquitination. It's also known as ubiquitination or ubiquitinylation. So you can pick and choose whatever you want. So basically what happens is that these proteins are labeled by these ubiquitin molecules, and they are destined for degradation by large complex proteins known as proteasomes. Okay, so these proteins function as factories. In other words, these proteins can be destined into lysosomes, where they also get degraded, and this is regulated as well. So basically, we talked about transcription and translation, and in between we have different levels of regulation. So we can have regulation of transcription by controlling the structure of the chromatin, converting it from heterochromatin into euchromatin, the relaxed DNA. We have control of transcription, like we saw before, with the like operon, with the enhancers and promoter proximal elements and so on. Now we have another level of regulation, that is RNA processing as well. Okay, splicing, for example, and so on. We have so we have synthesis of the mature messenger RNA. We talked about RNA transport as well. Okay, getting RNA from the nucleus to the cytosol. This step can be regulated. We have control of messenger RNA stability. We talked about that with the transferrin receptor. We talked about control of translation using different mechanisms of regulation. We talked about post-translational modification as well. Okay, so we're gonna leave this for biochemistry. Like, actually, we talked about it as well. Like phosphorylation. Okay, activating, inactivating protein by adding phosphates or removing phosphates. In biochemistry, we're going to talk about protein activity and how proteins can be regulated. We talked a little about that when we talked about GDP, GTP binding, phosphorylation as well. So the presence of inhibitors, that all lies under protein activity, controlling protein activity. And lastly, we also talked about protein degradation, and this can be regulated as well. So there are different mechanisms of regulation, and cells use these mechanisms for regulating different proteins, and different proteins can be regulated differently as well. Allah ma'akum.